All right, welcome back everyone. So in the last video, we were talking about enzymes and how they work and basically how they speed up reactions and so forth. So we're going to continue talking about enzymes, specifically how they're regulated. So move slide. All right, so let's start out by taking a look at how we can regulate an enzyme. So something could cause an enzyme to compete or to change the active site, I should say, and so that's called a competitive inhibitor. So a competitive inhibitor ends up binding to the active site of an enzyme, and that will basically force the substrate to move on. It can no longer bind to it. So you can see here's an example of an enzyme. Remember, enzymes have specific shapes where their active site has a very specific substrate that it can bind to. But let's say we have added in a competitive inhibitor. So that competitive inhibitor bound to that active site. So that means that the substrate can no longer bind to it. Okay, so that is an example of competitive inhibition. Okay. Another way that we can regulate enzymes or prevent them from catalyzing reactions is through non-competitive inhibitors. So in a non-competitive, well, the substrate's not competing with the, whatever the inhibitor is, but the inhibitor is binding to the protein at a different location that causes the active site to change its shape. So you can see how here we have our purple enzyme. That non-competitive inhibitor has basically bumped into it to a point that it has changed the shape of the enzyme's active site, preventing the substrate from binding to it. Okay, so that's a non-competitive inhibition. So here are some examples of competitive and non-competitive inhibitors with regards to enzymes. They could be certain toxins, so they can the toxins could mimic the substrate and bind to it. Poisons, same thing. Pesticides, they can prevent the substrate from binding to it. They could be a non-competitive or competitive inhibitor. And antibiotics can also be considered that. So any of these things can disrupt the function of an enzyme, either by binding to the active site itself or by changing the shape of the protein so that the substrate can no longer bind to it. Okay. So based on that information of competitive inhibitors versus non-competitive inhibitors, think about this question here. What determines whether enzyme inhibition is reversible or irreversible? So it has to do with the level of inhibition. Okay, so if that level of inhibition, say for example, a competitive inhibitor, it might just have a weak bond to the active site. And so once that bond is broken, now the active site is available for the substrate to bind to it. So that would be considered a reversible enzyme inhibition. It could also occur with non-competitive inhibitors. So if the non-competitive inhibitor, once it is no longer bound to the enzyme, if the enzyme's shape goes back to its normal shape, then it could be considered reversible. But if in order for the enzyme inhibition to be irreversible, there has to be strong bonds at the active site. So basically that inhibition mechanism is bound, pretty strongly to that active site, so nothing's getting in, or the shape is changed so much that it cannot be reversed. Okay, so it just kind of depends on the level of inhibition it is. Now we might think that this enzymes being disrupted could be always seems to be like a bad thing because it prevents a reaction from occurring. But that's not true. So we do want regulation of enzymes. We don't want certain reactions to occur over and over again in our body. So we need a way to kind of switch off and switch on. So we have certain genes inside of our body that encode for certain specific enzymes. So they can basically tell your body to make more of the enzyme if we need more of that reaction to occur, or to shut it down and not produce any more enzyme if we don't need that reaction to occur as often. Our genes can also regulate the activity of the enzymes. It can actually, cause it to turn on or off. So, excuse me. So when we have some sort of change in the enzyme's activity, whether it's going to prevent the enzyme from catalyzing the reaction, an inhibitory response, or it's going to stimulate the enzyme to catalyze reaction, that's called an I can't even pronounce it, alloteric regulation. Alloteric is, so basically this is going to either allow the enzyme to function or to inhibit its activity. So this could be an example of some sort of active site that ends up being altered when we don't want the enzyme to be active. And then when we do want the enzyme to be active, it returns to its normal state. So that would be an example of regulating an enzyme through an alloteric regulation. So that's some sort of regulatory molecule binding to the protein at one site, which can either allow the active sites to be active or the active sites to be no longer active. So let me show you what I mean here. So here's an example of an alloteric enzyme. So you can see in this enzyme, we have 4 different active sites, okay? And when we have some sort of activator, it's going to basically activate the enzyme's sites. So it's going to stabilize this protein molecule so that these active sites can receive substrates. We can also have instead of an activator, we can have an inhibitor. So you can see how that inhibitor binds to the enzyme in such a way that it changes the shape of the active site. So now we have a non-functional active site. So we have these alloteric activators that stabilize the active site for functioning, and we have the inhibitors that will change the active site so it's no longer functioning. So that will help regulate the enzyme's activity level. An example of an alloteric regulation that can actually amplify an enzyme activity would be something called cooperativity. So in cooperativity, you're cooperating basically by binding a substrate to one active site will cause a reaction to occur in a different active site. So here's our inactive form of our alloteric enzyme. And when we have a substrate is able to bind to that active site of one part of that alloteric enzyme, it's going to allow the other sites to be stabilized as well. So we have a substrate is cooperating or allowing for the change in the enzyme to allow for more active sites to open up. So essentially it's amplifying the activity of the enzyme. So it's once we have one active site utilized, that's going to amplify or open up for more reactions to occur. Now, sometimes we want an enzyme to function, and then we want it to shut off when we have enough of a product being formed. So we have something called feedback inhibition that can take place. So in this case, let's say we have this enzyme, and you can see how it says that the active site is available. So that active site, we're going to have a substrate bind to it. So that particular substrate is going to bind to it, and a reaction is going to take place. So that reaction is going to form some sort of end product that will then bind to another enzyme to have another reaction take place to form another end product, which will then bind to an active site of another enzyme to cause a reaction take place to form a final product. So in this case, the final product is isoleucine. Once we have enough isoleucine circulating, we don't need any more. So the isoleucine will either be used up by the cell, but if there's too much of it floating around, it will bind to the ulceratoric site of that enzyme. And that's going to, notice what happens, change the shape of the active site. So now this enzyme is not going to work anymore. And if that enzyme's not working, then you're never going to get that product. That's going to cause this product to occur, and this product, and this product, and so forth. So it's shutting down the whole pathway. So feedback inhibition, once we have enough of the product, it's going to basically change the shape of the initial enzyme that led to the reaction. So we're going to be focusing on cellular respiration in Chapter 7. And in Chapter 7, we're going to focus on the site of cellular respiration, which is the mighty mitochondria. So you might be familiar, with going back in Chapter 4 when we talked about the mighty mitochondria. And what's interesting about this mitochondria is that it's going to be undergoing this very important chemical reaction in order for us to get enough ATP, so enough of that ATP to form. And so to aid this process, of course, we're going to have enzymes involved. So enzymes, they can be within the matrix, so they're involved with one stage of cellular respiration. They're also embedded in the inner membrane, so they're involved with another stage. And we'll get into all of that next in the next chapter. But just realize that, these enzymes, they're all over our body, and they are catalyzing all sorts of reactions in our body. Okay. So let's finish our enzyme talk with kind of a review of some examples as well as that feedback inhibition that we just discussed.
Have you ever noticed how, when learning more about a science topic, we can get so interested that we start to discover favorites? Favorite dinosaur? I've found quite a few opinions there. Favorite animal? I'm a little obsessed. Favorite flower? Well, sometimes a little too obvious. Of course, you can always get a little more unique. Favorite protist? Favorite amino acid? Your favorite nitrogenous base? I'm a little saddened that no one has yet asked me those three above, nor have they asked me what I've been waiting to be asked, and yet have often volunteered. What is your favorite enzyme? ATP synthase is mine, but it's a hard choice because there are a ton of fascinating enzymes. We already have an intro video about enzymes, but this video topic is about to go into a little more. Enzyme examples, cofactors and coenzymes, inhibitors, and feedback inhibition. Enzyme examples are important because without learning about some real-life examples, it's kind of hard to connect to how important they are to living things. Let's start with some fascinating enzyme examples in the human body. A great place to focus on digestion. Many enzymes focus on breaking down certain biomolecules. You remember the 4 biomolecules: carbohydrates, lipids, proteins, nucleic acids. These biomolecules are found in your food. You've got to break those biomolecules down in digestion. Amylase is an example of an enzyme that breaks down carbohydrates, and one action location is in the mouth. Amylase can break the glycosidic linkages in starch, which is a large carbohydrate, into smaller carbohydrates. Lipase is an example of an enzyme that breaks down lipids, and one action location is in the small intestine. Lipase can break the ester bonds found in triglycerides, which are a type of lipid, into their building blocks: fatty acids and glycerol. Pepsin and trypsin are some examples of enzymes that break down proteins. An action location for pepsin is in the stomach, and an action location of trypsin is in the small intestine. These enzymes break the peptide bonds in proteins. Pepsin breaks down proteins into peptides. You even have nucleases that break down nucleic acids. That's DNA and RNA. And yes, DNA and RNA can be found in your food. Nucleases break down phosphodiester bonds found in connected DNA and RNA and helps break them into nucleotides. By the way, once all of these biomolecules are broken down into smaller pieces, there are even more digestive enzymes that can break them down even more. But it's not just digestion. You can name any human body system, excretory system, respiratory system, circulatory system, and you'll find there are enzymes involved. These enzymes might break down or build up the substances that they act upon, which is needed by different body systems. And while you're at it, realize that enzymes are not just for humans. Enzymes are found in all living organisms, like this Venus flytrap here. Beyond the three domains of living organisms, you'll even find some types of enzymes in viruses. Okay, so moving on. What's next on our list? Oh, yes, cofactors and coenzymes. So, just a quick review from our main enzyme video. This is an enzyme here, and we have our active site, the substrate that could fit in the active site for the enzyme to act upon. It's an enzyme substrate complex when the substrate is bonded in this active site. And there is likely induced fit, where the active site actually changes its shape slightly for an ideal fit, an enzyme substrate hug. But the thing is, enzymes often don't do it alone, and they tend to have cofactors, which tend to be inorganic, like zinc or iron, or coenzymes, which tend to be organic, like many vitamins. Cofactors and coenzymes may be permanent or temporary, which often depends on how they're bonded. A cofactor or coenzyme may sit in this active site here. It helps the bonding of the enzyme and substrate so that it can work at its best, and sometimes it won't even work at all without them. An example of a cofactor or coenzyme? You know when we've mentioned DNA polymerase, an enzyme in DNA replication? It often has a zinc ion as a cofactor to help. There are also inhibitors. They might be reversible or not reversible. A lot of that depends on how they bond. They may be competitive or non-competitive. Competitive inhibitors tend to sit right here in the active site, blocking a substrate from being able to bind. The substrates are competing with the competitive inhibitor for binding. Non-competitive inhibitors bind to some other area of the enzyme, somewhere that isn't the active site. This can be referred to as an allosteric site, a site on the enzyme that isn't the active site. When the non-competitive inhibitor binds on the allosteric site, the enzyme has a shape change. If the substrate still binds, the enzyme's active site might not function well, or at all. You know, inhibitors might sound like a bad thing, but inhibitors can be harmful or they can be helpful. Some examples. You might remember DDT from our biomagnification video. It can act as an inhibitor for certain enzymes in the human body and can cause health issues. But understand that our body also uses inhibitors all the time to control a lot of biological processes. It's called feedback inhibition. This is a hypothetical example here, but let's say that enzyme 1 converts substrate A to intermediate B. Then enzyme 2 converts B to C. Then enzyme 3 converts C to a final product D. These enzymes are all working in a pathway here. But what if you have enough product D now? It's the ultimate product, but you have enough, and it's wasteful to keep producing it, or maybe it's even harmful to make too much. Well, product D might also be a non-competitive inhibitor for enzyme 1, stopping enzyme 1 from working, which will stop the process. Again, this could be reversible, or it could be that more enzyme 1s are produced in the body if this needs to start up again. So to recap, we explored some real-life enzyme examples, talked about cofactors and coenzymes, inhibitors, and feedback inhibition. But one last thing. We're big on asking why. What is the significance of learning this? So we know that enzymes are ubiquitous, that word, and they are critical for understanding biological processes. They're also important to understand because many of the medications we use to treat diseases and disorders act on enzymes. One example, high blood pressure. It can be harmful to your organs and blood vessels. One class of blood pressure medications is the class of ACE inhibitors. ACE inhibitors work by blocking angiotensin-converting enzymes. When inhibited, these enzymes can't convert angiotensin to angiotensin 2. Angiotensin 2 can be involved with raising blood pressure, so By blocking the enzyme that helps produce it, it can lower blood pressure. There's some more complexity with this to explore. By the way, it's not just the human body. Medications can act on enzymes and microbes that make us sick. For example, penicillin. Penicillin is an antibiotic, but it works by inhibiting the enzyme transpeptidase, which prevents many types of bacteria from being able to build their cell wall. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so again, I like that video because it gives us, a good overview of what um, enzymes do, and then what happens to regulate the enzymes with those competitive, non-competitive inhibitors and feedback inhibition and so forth. So that was kind of a good summary, as well as applying it to some of the conditions in our body. Like we have certain drugs that can help prevent an enzyme from working that would contribute to high blood pressure. So basically we're done with chapter 6, but we're going to, instead of just stopping there, we're going to start getting into a little bit of chapter 7. And the reason being, chapter 7, cellular respiration is a very complicated process, and a lot of students, need some extra time working on it. So we're just going to introduce it today, and then, give us a good overview. And we're going to apply some of the information we learned from chapter 6 into this beginning part of chapter 7. So we'll talk about those reactions again. And so that will give us a good foundation for when we talk about exactly what takes place more specifically in cellular respiration and fermentation. So we're only going to be basically looking at this first part, catabolism. So remember, hey, we talked about catabolism at the beginning of, or the last lecture. So this will be relevant and a bit of review, okay? So in order to start out our talk about cellular respiration, we need to realize, well, where it comes from. Okay, so why do we need cellular respiration, and where is where does it originate from? So we can actually draw all of the energy that you require to do all of your functioning. That energy at one point was sunlight. Okay, so that was actually light energy from the sun. So the energy that you're using right now to, process your thoughts and everything, that we could classify that as solar energy at one point. But how did it get into your body, and how is your body using it? Well, that's where basically the food chain comes in, if you remember that from elementary school. So we have our plants. Our plants are going to do the process of converting that light energy from the sun into a usable form of energy for us, which is chemical energy, food. So here's an example of how that happens with photosynthesis. So the plants, they're going to be inside the chloroplasts of their plant cells. They will be catalyzing that photosynthesis reaction with the help of the light energy from the sun. So we're basically, that plant is taking the carbon dioxide, which it takes in from its surroundings through these little pores in its leaves called stomata, and then it absorbs water from the roots. So those two reactants are going to go into the chloroplast of the plant cell, where the light energy from the sun will drive that chemical reaction to form glucose, C6H12O6, and other types of organic molecules. And as a byproduct, it's going to produce oxygen gas, okay? So now there's that organic molecule, glucose, or whatever it is, fructose, some sort of organic molecule. That's the chemical energy now. So we have gone from taking light energy and converting it, now it's in a chemical form that our body can use. So plants, they can use their own chemical energy that they produced for cellular energy and convert it to cellular energy through cellular respiration. Because plants, they need to do things too. Okay, so it's not just animals that undergo cellular respiration. Plants, they do it too. They have mitochondria, just like we do. And so they're going to take that chemical energy and they're going to convert it into cellular energy, ATP. So they're basically self-sufficient. They can absorb energy from the sun to generate, convert it to chemical energy, and then they can undergo cellular respiration to generate cellular energy. We don't have the ability to absorb light and convert it to chemicals. So what do we need to do? We need to eat. So we eat those nutrients either directly by eating a plant or by eating an animal that ate the plant. And so now we have those organic molecules in a chemical form that can then be used in this reaction called cellular respiration in order for us to produce ATP, or cellular energy. So that goal for both plants and animals is to create cellular energy so that we can do work. Okay? So there's this, here's a misleading statement here about plant cells. So plant cells perform photosynthesis, animal cells perform cellular respiration. So a lot of students get into that misconception that, plants do photosynthesis, animals do cellular respiration. Remember, plants also do cellular respiration. Okay, we need chemical energy to convert to cellular energy. So do plants. Okay, the only difference is they can make their chemical energy through photosynthesis. They don't have to eat it like we do. Okay, so they still need cellular energy. They need that type of energy to power their cell parts and what plants do. Okay, so plants do both. We only do one. Okay, so let's go back to what we talked about at the beginning of chapter 6 with catabolism. So remember, catabolism is that process where we're going to break down a large molecule into smaller components. And if you recall, that is an example of an exergonic reaction because the organic molecule, that complex organic molecule, has a lot of stored energy. Okay, whereas the products will have less energy. So this will be a negative free energy situation. So it's an exergonic process where in order to break apart the organic molecules, energy will actually get released. So cellular respiration is going to be an example of that process where we're going to take organic molecules, such as glucose, and we're going to release energy from it in the form of ATP. Okay, so that's that energy that we're trying to acquire or basically convert to. So this example of cellular respiration is a catabolic pathway. And the reason why it's a catabolic pathway is because we're going to be taking forms of energy, in this case it's electron transfer, to be able to take ADP plus phosphorus to create that ATP, that energy source. Okay. So cellular respiration, exergonic, it's going to involve electrons, and it is a catabolic process. Okay, so so far we're using some terms here that we talked about in the last presentation. In addition to cellular respiration, that's not the only way that cells can get ATP. There's other ways. So some cells can undergo a form of what we call anaerobic respiration called fermentation. So fermentation is basically where we take the organic molecules, that chemical energy, but we break it down in the absence of oxygen. So oxygen is not involved. Okay, so our cells can do this if there's not enough oxygen. Certain fungi can do this, certain protists can do this, certain bacteria can do this. Oops. Sorry about that. Back. There we go. So we're going to look at fermentation as well. Just realize that fermentation is another way that we can get that ATP, that cellular energy. But what's interesting about it is it doesn't it doesn't give us as much. Okay, so it's a little bit inefficient. Whereas aerobic respiration is where we're going to basically consume those organic molecules. I don't know why it got broken down, but and process them in the presence of oxygen. So think aerobic, oxygen present. Anaerobic is without oxygen, so one form of that, well, there's a couple different forms of fermentation. Okay, so all of these processes are catabolic because we're taking big, large organic molecules such as glucose and we're breaking them down and using that energy and releasing it. So let's take a closer look at that catabolic pathway. This is an example of cellular respiration. This case is aerobic respiration, okay? Cellular respiration, aerobic respiration. So this is the one that we're going to be focusing on. So the reason why it's aerobic, because oxygen is present. So we're going to break down this glucose molecule with the presence of oxygen. And by breaking it down, it's going to release that form of energy that our cells need to work, ATP. And you can see the byproducts of the process. So carbon dioxide and water are also produced along with heat. Okay, so this is going to be generating that ATP cellular energy that our bodies are seeking to do work. Anaerobic is basically the same process, but without the presence of oxygen. So certain bacteria, certain protists, certain organisms can strictly undergo anaerobic respiration and still produce quite a bit of quantity of ATP. Typically what happens is they use some other energy carrier. So this oxygen is used as an electron carrier. We'll talk more about that later. But they have some sort of some other molecule, inorganic molecule instead of oxygen. And then there's other forms of anaerobic respiration where there's there's fermentation. So basically you're just breaking down the sugar without any oxygen present. So that one's not going to produce as much ATP. All right. So as far as this process, when we're just looking at cellular respiration, aerobic cellular respiration, it's a fairly efficient conversion from the energy stored into glucose into converting it to this usable form of ATP. So it's about 36 to 40% efficient. So you might think, well, that doesn't seem like much. I mean, it's only up to maximum 40% efficient. So that means 60% of that energy gets converted into heat. Okay, that doesn't sound very efficient. But it is if we compare it to other reactions. So for example, your automobile, only about 25% efficient, okay, in taking that gasoline and converting it into movement by your vehicle. I'm not an automobile person, so I don't know the mechanics of it, but the conversion from the form of energy, the chemicals, into the actual movement of your vehicle with that energy is only about 25%. So the other 75% is lost as heat. So you could think, hey, your cells are more efficient than an automobile. Anaerobic processes, especially when we come to fermentation, they generally are not energy efficient. Okay, so we can see the conversion factor here where it's only about 2% efficient. So the other 98% gets lost as heat or is unusable. It will not produce that ATP. So it can produce a little bit, but not nearly as much as aerobic respiration. All right, so energy intake, 75% of intake used in life-sustaining activities. Okay, so basically you're going to be taking in that energy in the form of chemicals and then converting it into ATP about 75% of the time. Okay, so we're using it to maintain our life-sustaining activities. So the rest of that 25% is basically used for other things. Okay, so most of that energy that we burn in calories is just so that you can breathe, so that you can, process thoughts. You can have your neurotransmitters transmit and all that fun stuff. So the majority of your energy that you're consuming is just going to just keep you alive. The other 25% is what you are utilizing for extra activities such as moving around, doing exercise, talking. Okay, so all of that is not required for survival, but you just, that's where that extra energy can be used for. Okay. All right, so this process, this catabolic reaction we're about to explore with cellular respiration, we're going to need to look closely at these special high energy molecules that actually transport electrons to one area of the mitochondria into another. So these are called energy carriers, and they carry the electrons, or electron carriers. And so they're going to be involved with something called an electron transport chain, which is where we're going to actually see the energy from this transport chain as it undergoes these changes is going to help provide the energy to drive the ATP, or the ADP plus the phosphate group to form ATP. So throughout cellular respiration, we're going to see how we have these energy carriers are carrying electrons to this final part of cellular respiration, which we call the electron transport chain. So you're going to be paying close attention to these electron carriers. The initial molecule that is used is, we actually have two of them. We have something called NAD plus and as well as FADH. So what ends up happening when we have a reaction, okay, so this NAD plus, it's going to basically, it's going to end up gaining electrons, and as it's gaining electrons, it changes form. Okay, so it goes from NAD plus, and it's gaining 2 electrons, to something called NADH. So this NADH acts as an electron carrier. In fact, it's carrying 2 electrons. And that's going to be important because we're going to need enough energy to drive the final reaction of cellular respiration with the help of those electrons. Okay, so energy, these electron carriers now are high energy molecules. They're carrying that electron, that form of energy. Okay. Another molecule that you're going to be noticing throughout these chemical reactions is called FADH. And so FADH is another example of an electron carrier. So when it has electrons added to it, it actually forms something called FADH2. Okay, so that will be an electron carrier that can carry more electrons to the electron transport chain. Okay, so FADH and NAD plus are going to be transferring some of the electrons from some of these earlier reactions to allow for us to get enough energy to create ATP at the end. Okay. Now these type of reactions, you'll notice we have these terms called oxidize and reduction. So we're going to talk about that in a second. Okay, so if you're kind of confused what this means. But let's take a closer look again at NAD+, so that's one of our electron carriers. Okay, so if we see NAD+, you notice it is going to be accepting electrons. And when it accepts electrons, it converts to NADH. So let's say that this NAD+ is out in the outside of the mitochondria. It's in the cytoplasm. When it gains those electrons, okay, it's going to be moving as NADH into the mitochondria, into a special area of the mitochondria where it can release these electrons. These electrons will then be transferred through this, what's called the electron transport chain, which will basically allow for enough energy to take that ADP and the phosphate group to form ATP. So it's going to help synthesize ATP. So we're going to get that energy, that cellular energy, through basically this step-by-step process. So that's why I'm introducing it now, because we need these electrons carried by an electron carrier to power this process. Okay, so this is going to help us synthesize the ATP in the end. And throughout the cellular respiration reactions, there's multiple steps. You'll see we're gathering these electrons that will then be carried to the electron transport chain. Okay. So remember, how do we make ATP? So ATP, basically we need to undergo a reaction, right? So we need to take that ADP plus the phosphate and bring them together. So there's actually an enzyme that's involved. It's called ATP synthase, and it is found in the mitochondria. And it's going to basically allow this reaction to take place. Okay, so this is going to occur with the help of those electrons. Okay, so they're using electron energy to kind of drive this reaction to form that ATP. Okay, so enzymes transfer the phosphate group from the ADP using the electrons that were transferred to the mitochondria. So we call that a substrate level phosphorylation. We'll talk more about that. All right. So we talked about those electron carriers that will be important in the last step of cellular respiration. Again, we'll get into more details next week. And then we talked about how ATP is going to be used and how it's going to be formed using those electrons. So we're going to get into basically focus in on those electrons themselves. So when we have a reaction that ends up gaining electrons, it's called a reduction reaction. Okay, so reduction reactions gain electrons. Oxidation reactions lose electrons. So understanding this, we see that oxidation and reduction reactions are a form of coupled reactions. So we actually have one occurring that's going to drive the other one. Okay, so that's called a redox reaction or reduction oxidation reaction. So here's an example of that. Hey, here's an ionic bond, right? So we have our sodium. If you recall from, wow, I think it was like the first week of school, but we talked about the ionic bonds and how sodium has just one electron in its outermost valence shell. And chlorine has 7. So chlorine wants to gain an extra electron, whereas sodium wants to lose that extra electron. So if sodium loses that electron, becomes a positive ion, that would be an example of it being oxidized. It's losing an electron. So that's the oxidation part. The chlorine is going to pick up that extra electron. So it's going to gain that electron. So that's the reduction part. So you can see how the oxidation is occurring. That's at the same time as the reduction. And so that's why we call it a redox reaction. Right? So I'm going to show you, I don't know if we did do a crash course video earlier with the cells, but this one is going to get a little bit more technical than what we need for the purposes of this class because it's based on a chemistry course, but it does do a good job explaining redox reactions. So we're going to watch that.
Atom to atom is a redox reaction. That oxidation is the loss of electrons, and that reduction is the gain of.
Let's start from the beginning. I don't know why it started way back there. All right, here we go.
For atoms to form molecules, and when that happens, A tremendous amount of energy can be exchanged in the process. But not all chemical reactions involve electrons changing hands. Acid-base reactions, you'll recall, are more about swapping protons. But because electrons are the real coin of the realm in chemistry, the most important reactions that take place on Earth involve the transfer of one or more electrons from one atom to another. These are redox reactions. Redox, which is a portmanteau of reduction oxidation. But what's up with those words? You know what reduction is? Making less of something. And then oxidation maybe has something to do with oxygen. Well, sometimes, but not always. These are actually super terrible choices for what is actually happening in redox reactions, but we are stuck with them. Reduction is when a substance gains electrons. Yes, it gains, which is the opposite of what the word reduce means. Fantastic. And yes, sometimes I want to punish the people who named these things so inaccurately, but they didn't know any better and they're all dead, so we can't do anything about it. Protochemists would make pure metals by heating or smelting their ores. And they noticed during the smelting that these substances would become lighter. So I guess it's not crazy that they decided to say that these substances were being reduced. Our old French friend Antoine Lavoisier figured out that this was because oxygen gas was actually leaving the compound, making it lighter. What he didn't know was the actual chemistry involved. Oxygen is, unsurprisingly, the quintessential oxidizer. It pulls electrons off of one molecule to make itself more stable. But if you heat it up enough, it gets all energetic. Today, we understand that oxidation and reduction are all about electron transfer. So you might think that we'd rename them, and some chemists have tried producing terms like electronation and de-electronation. But once a set of terms is decided upon and used for a while, it's pretty difficult to uncreate it. So we're stuck. To keep these seemingly nonsensical names straight, I rely on the phrase oil rig. Oxidation is loss of electrons, reduction is gain of electrons. We just gotta know this stuff, because it's everywhere. When your cells convert sugar into energy so you can move and breathe and think, that's redox. When plants photosynthesize sunlight into food, that's redox. The battery powering your laptop? Redox. Fire? Also redox. Since electron swapping is the name of the game here, when you study redox reactions, it's important, critical, absolutely essential to keep track of the electrons. Think of them as dollars or pesos or pounds or euros. In any transaction, one person is going to gain them and the other is going to lose them. And to stay on top of things, you have to keep tabs on who's ahead and who's behind. Atoms are fond of sharing electrons, though, forming covalent bonds, so sometimes keeping track of where they are and where they're going to end up isn't quite so simple. Let's think of every covalent compound like a marriage, though it's going to be a weird marriage because there might be 6 people in it, sometimes the same person several times, without commitment, no commitment, and also no emotions. Don't think too much about it. Like in a marriage where money gets shared, covalent compounds share electrons. The trick is figuring out who gets the cash when the vows break. So we've created a useful little system assigning electrons 100% to atoms that are actually at the moment sharing them. The number that we assign is the atom's oxidation state, or oxidation number. Even though we are of course aware of covalent bonds and the sharing of electrons, the processes are easier to follow if we imagine that the atoms are already splitting up the bank account, as if they were in an ionic or non-sharing bond. So an atom's oxidation number is basically what its charge would be if it actually owned all of its electrons exclusively, like the newly minted bachelors that they may become. So to figure out those oxidation states, or oxidation numbers, we have some simple rules for some atoms. First, the oxidation number for any element by itself, whether it's monatomic or diatomic or polyatomic, like an atom of calcium, or a molecule of H2, or an even bigger molecule of sulfur S8, the oxidation number is 0. Atoms, by definition, do not have a charge. If they had a charge, they would be ions, and if they're sharing with themselves, they share equally. Second, for a monatomic ion, basically a charged atom, it's simply the size or number of its charge. So the iron 2 in Fe2+ has an oxidation state of +2, while the chloride ion is -1. Third, oxygen, which is unsurprisingly all over redox chemistry, almost always has an oxidation of -2, unless it happens to be in a peroxide molecule like hydrogen peroxide. Fourth, hydrogen is +1, and fifth, fluorine is -1, as are all the other halogens most of the time. Pretty much unless they're bonded to fluorine or oxygen, because fluorine and oxygen are so bad that they can make anybody's oxidation number positive, if you know what I mean. And those are the rules. That's all you need to know. Now, the total of all the oxidation numbers of all the atoms in a neutral compound will add up to 0, like water with one oxygen with a -2 oxidation state and two hydrogens at +1, and voila! The neutral compound has an oxidation number of 0. A polyatomic ion, on the other hand, has to work out to have an oxidation state that matches its charge. So SO42-, the sulfate ion, has 4 oxygens for a total of -8, but we don't have a rule for sulfur, so I guess we just give up and walk away. Who cares anymore? No! We use like 3rd grade algebra because we have to end up with an oxidation number of -2 for the whole compound. We know that sulfur, in this particular compound, has an oxidation state of +6. But sulfur's oxidation state isn't always +6, and that's why we don't have a rule for sulfur, or a lot of other elements for that matter, because oxidation states of most elements change depending on what they're bonded with. Now we can use this same logic to figure out what happens when these compounds interact in redox reactions. Molecular divorce courts of electrons changing hands, being haggled over and traded, with some players taking big profits while others lose nearly everything. Them's the breaks. Let's start out with a simple example. A chemical reaction that I believe has saved more lives than any other in the history of chemistry. Created by a war criminal to blow people up during World War I. The Haber process. The Haber process removes the ultra-stable elemental nitrogen from the air and combines it with hydrogen to form NH3, ammonia, for use in bombs and also in fertilizer, increasing the carrying capacity of the Earth by billions. Nitrogen in the air exists as elemental diatomic nitrogen, and hydrogen likewise is also diatomic H2. So we know that starting out, all of the atoms have an oxidation state of 0. The product of the reaction, ammonia, is a neutral compound with 1 nitrogen and 3 hydrogens. The hydrogens each have an oxidation state of +1, remember the rules, so nitrogen must have an oxidation state of -3. Nitrogen thus gained electrons, its oxidation state went down, and so it was reduced. So at least when talking about what oxidation states are doing, the word reduced makes sense. Hydrogen lost electrons, the oxidation state went up, and it was oxidized. Now this is a pretty simple equation to balance, but redox equations can be a huge headache sometimes because of the number of individual atoms involved. So we often have to balance them in half reactions. So even though we don't really need to do the half reactions because this is a pretty simple equation, we're going to do them anyway, just because it's an example that's simple to start with. So start out with the nitrogen getting reduced. We have N2 with an oxidation state of 0 becoming NH3 with an oxidation state of -3. First, we balance the number of nitrogens, then add the number of electrons that we need to have there be the same number of electrons on each side. Do the same with the oxidation half of the reaction, and then combine them to get your whole reaction with the electrons canceling out. Now yes, maybe that seemed like an unnecessary step, but allow me to show you a more complicated example that will prove how necessary it may be. In this flask is silver diamine. We're going to use some redox chemistry to get the elemental silver out of it nice and clean and shiny, and it's not going to be no simple Haber process. The silver diamine is going to react with an organic aldehyde, any aldehyde actually. The business end of the aldehyde is the CHO end. The R in organic chemistry is a symbol for some organic group of atoms, and in this reaction those atoms don't matter. The silver diamine reacts with the aldehyde and hydroxide, creating carboxylic acid, ammonia, and water. First, let's assign ourselves some oxidation states. The silver is in a complex with 2 neutral ammonias that are going to remain unreacted throughout the equation, so we can treat them like a single species with an oxidation state of 0. Since the silver diamine has a charge of +1 and the ammonias don't affect that, silver's oxidation state must also be +1. The aldehyde has 1 hydrogen at +1 and 1 oxygen at -2, but is neutral overall, so the carbon must be +1 as well. The hydroxide ion is simple, -2 for the oxygen, +1 for the hydrogen, and an overall charge and thus oxidation state of -1. On the reactant side, the silver is now atomic, so its oxidation state is 0. The carboxylic acid has 2 oxygens and 1 hydrogen, so the carbon now has an oxidation state of +3. NH3 remains at 0, and the hydrogen and oxygen of water also haven't changed oxidation states. So silver's oxidation state decreased or was reduced from +1 to 0, while carbon was oxidized from +1 to +3. Half reaction time. Silver was reduced, gaining 1 electron, forming elemental silver and ammonia from silver diamine. The aldehyde was oxidized, forming carboxylic acid and requiring 2 electrons. With the help of those electrons, we know that at the very least we have to double the reduction half of the equation entirely in order to get the right number of electrons on both sides. We do that, and oh god, that's good stuff. And we combine them together for a perfectly balanced redox equation. And now, watch me take those electrons and turn them into money. And there you have it, folks. That is pure silver coating the inside of the flask. Thank you for watching this episode of Crash Course Chemistry. If you were paying attention, You learned that any reaction where electrons move around from atom to atom is a redox reaction, that oxidation is the loss of electrons, and that reduction is the gain of electrons.
Okay, so like I stated before, oxidative states, you don't really need to know those. We're gonna kind of just focus in on the redox part, but now you should understand what a redox reaction is and what reduction means versus oxidation. So remember, he used the acronym oil rig. So oil rig, oil or oxidation is losing electrons, and then redox or rig, sorry, the R stands for reduction and then is gaining electrons. So that might be helpful in understanding what a redox reaction, which one's losing the electrons, which one's gaining the electrons. Okay, so let's bring this back to our cellular respiration equation. So this is the cellular respiration equation that you will become very familiar with over the next week. So we're going to start out with the glucose molecule. So that glucose molecule is C6H12O6. Okay, so this is a complex molecule. And it is going to end up losing an electron, or in the form of basically it loses the hydrogen atoms. So in this case, since it's sorry, it's not losing, it's gaining electrons. So basically the loss of the hydrogen atoms is in the form, it's going to become oxidized. So oxidized, we lose that electron, so that's the oxidation reaction that's occurring. That oxygen on the reactant side is going to undergo a redox reaction. And by undergoing that redox reaction, it's going to gain the electron, or what we call a hydrogen atom in that sense. So the glucose becomes oxidized, the oxygen becomes reduced. And so we can see how the electron transfer is taking place. So as hydrogen with its electrons are transferred to the oxygen, energy is then going to be released, and that can be used in ATP synthesis. So the fuel, the glucose, will be oxidized, the oxygen will be reduced. Now, what happens when that glucose is oxidized and it loses that electron? Okay, so as it's losing that electron can then be added to that electron carrier we talked about, NAD+. So we think of that NAD+ as an oxidizing agent. So it's going to take that electron that was lost through that oxidation process. And that will then bind to the NADH to form NAD, or the NAD+ to form that NADH. So the way it does that, basically we have our NAD+, and then that NAD+ will undergo a reaction with the help of an enzyme. You don't really need to memorize it, but that rearrangement of that reaction is going to create NADH, where 2 electrons are going to be added to form this NADH molecule along with a hydrogen ion. So each NADH molecule is going to represent that stored energy that's tapped in the synthesis of ATP, because it carries those electrons. Once those electrons are lost, okay, then it will basically return, it'll undergo its own reaction to return to NAD+. Okay. So let's take a closer look. Here is an actual NAD+ molecule. It's very complex, and you don't even need to know the full name of it. But we have the NAD+, so this is in its oxidized form. So it's going to be gaining electrons. So in gaining electrons through some sort of oxidation reaction, those electrons will be then added. So now we have the reduced form, which would be the NADH. So it's gained those 2 electrons. So now that NADH can then release those electrons. When it releases them, it's oxidized back to its NAD+ form to gain more. Okay? Now, all of those electrons, they're going to be basically going to be involved in this electron transport chain. So that electron transport chain is going to take all of those electrons that the electron carriers had dropped off, as well as these protons that get pumped through, and that is going to help basically release a bunch of energy, and that energy is going to create that ATP. Now the final acceptor of the electrons, so once the electrons are transferred through, okay, they don't go away, they're going to be accepted by oxygen molecules, and that's going to be used to form H2O with the hydrogen. Okay, so the oxygen is this final electron acceptor. And if this is confusing, again, we're going to go kind of more into it next week. But I do want to just introduce the stages of cellular respiration, and then we'll watch a video and then we'll call it a day. So we can take cellular respiration and we can break it down into 3 processes. One of them occurs outside of the mitochondria, the other 2 occur inside the mitochondria. So the first stage is called glycolysis. So in glycolysis, we're going to be taking glucose, so think glycolysis, glucose, and we're going to break it down. So we're actually going to break it down into 2 molecules. If you remember, glucose was C6H12O6, so there's like 6 carbons. We're going to break it down into 2 molecules that each have 3 carbons. So we call that pyruvate, those 2 molecules. So that's the first step. We've broken down glycolysis. And in order for this step to occur, we actually require a little bit of energy to get things started. But we end up producing a little bit of ATP in the process, along with getting those electrons transferred to electron carriers that get carried into the mitochondria. Those pyruvate molecules will then be oxidized. Okay, so what does oxidation mean again? So remember oil rig, we're going to lose electrons. So that pyruvate oxidation is going to occur in the mitochondria, and it's going to undergo a series of reactions known as the citric acid cycle or the Krebs cycle that will break that pyruvate molecule down even further. And then and that will produce a little bit more ATP, but mainly it's getting all of these, since it's an oxidation reaction, we're losing a bunch of electrons that then get attached to our electron carriers and then transported to the final stage of cellular respiration, which is also known as oxidative phosphorylation or the electron transport chain. So remember oxidative phosphorylation, that's when the ATP becomes phosphorylized, it becomes, it gets its extra phosphate group, right? And then this term oxidative just means that the oxygen will be the final electron receiver. And then we've talked about that electron transport chain where all those electrons are ending up, they're going to allow this process to take place. So this is where the majority of ATP synthesis is going to occur in cellular respiration, about 90% of it. And it occurs in the inner membrane of the mitochondria. Okay, so a lot of information. We're going to break it down in detail next week. But let's just do an overview of cellular respiration so we can kind of add all those terms that we talked about with the electron carriers and electron transport chains and all that, put it together so it kind of solidifies in our memory. Okay.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Okay, so I know you got lots of ATP right now functioning, trying to figure out what everything was talking about in the video. Don't worry, we're going to go through it specifically in detail next week. For now, I do just want to point out for this part of the lecture quiz, you should know, basically the redox reactions, what those are, and then how they relate to this cellular respiration thing that we're going to go into more detail with. And then just recognize that in your textbook, since it is such a complex process, we do divide them into different stages, or cellular respiration into 3 states. So glycolysis is always labeled as this blue stage throughout the chapter. Pyruvate oxidation and citric acid cycle or Krebs cycle is orange, and then the oxidative phosphorylation, where we have the electron transport chain, and chemoosmosis that occurs, you'll see as purple. So let's start out by just kind of simplifying everything for the purposes of our lecture quiz for this week. So we're not getting into the nitty-gritty details. but we do need to kind of understand the phases a little bit. So with cellular respiration, we have those 3 phases: glycolysis, the Krebs cycle or citric acid cycle, and the oxidative phosphorylation electron transport chain. So let's start with glycolysis. This occurs outside of the mitochondria. So you can see the mitochondria right here. So we're in the cytosol. This is where we're going to take that molecule of glucose that we've consumed, and we're going to break it down into 2 molecules of pyruvate. So this process, it's going to end up the product being pyruvate, which will then be managed to make its way into the mitochondria, will be further broken down. But this process in itself produces a little bit of ATP. And then it also is going to, since it's going to be an oxidation reaction, glucose will basically essentially lose those electrons that then get attached to our electron carriers to form NADH. So that's going to enter the mitochondria. Next, we have the second stage, which is kind of divided into 2 parts. We have basically pyruvate oxidation. So that pyruvate that's made its way into the mitochondria will be further broken down with the help of acetyl coenzyme A. A little bit of carbon dioxide got released. And then now it's ready to enter this citric acid cycle or Krebs cycle. I think citric acid is the more common term used these days. In the past, it was Krebs. So basically in this cycle, we're going to further break down the original glucose molecule. And in that process, we're going to be stripping all of these electrons from it. So those added electrons get added to NADH or to NAD plus to form NADH and to FADH to form FADH2. So now we have a bunch of electrons going to the last part, the inner membrane. A little bit of ATP is produced in that process. So we've gone from glucose to now we've basically broken down the glucose and extracted a bunch of electrons from it and a little bit of ATP. And then the final stage is this where the bulk of ATP synthesis occurs. So that's where we're going to end up stripping the electrons from the NADH and FADH2. Okay, so these guys will lose their electrons and become oxidized. And then we're going to use those electrons to basically pump protons or hydrogen ions across a membrane. And it's going to end up causing so many protons on one side of the membrane that we have this concentration gradient that's formed. And that's going to power that enzyme, that turbine, that's going to basically allow for the protons to pass through again, providing the energy for ATP to be phosphorylized, or ADP plus protein to, or proton to phosphate group to be forming ATP again in its oxidative state. So we're going to basically be getting the end state of ATP with all of its energy. Okay, so we'll go through this in detail next week, but for now, just make sure you kind of understand there's 3 phases of cellular respiration. All of them produce ATP, but the bulk is at the end. Okay? All right, so this is it for chapter 6 and the beginnings of chapter 7. We'll go into more detail next week with chapter 7. Okay, so if you have any questions, feel free to email me or post in the discussion boards. Otherwise, I'll see you at the Zoom meetings.